Okay, good morning, everyone. Thank you for attending today's webinar. My name is Mackenzie Woodman. I'm the Remote Sensing Application Specialist here at Spectral Evolution. And today we're going to be going over remote sensing methods, particularly focusing on field reflectance measurements. So we'll go through some of the methodologies. They'll be very generalized because it has to be for every application. It will have specific different procedures, but we'll speak generally on procedures for different sampling techniques, as well as talk about some of the pitfalls and things to consider when taking field reflectance measurements. So first I'll introduce Spectral Evolution. Spectral Evolution is the leading manufacturer in high quality UV-VIS-NIR spectrometers. Our typical range for spectrometers and spectroradiometers is 350 nanometers to 2500 nanometers. We've been around for 20 years. We were established in 2004. And we're located in the United States, in the Boston area in Massachusetts, so Haverhill, if anyone's familiar. Our typical applications are in hyperspectral, remote sensing and vegetation, mining and geology, as well as photonics and calibration transfer. So the first thing I want to do is go over what is spectroscopy to give you a better understanding of how these instruments operate and what you're really measuring in the field. So spectroscopy is the study of the electromagnetic spectrum as emitted or reflected by various materials. The electromagnetic spectrum is a representation of electromagnetic radiation, and they are defined by their wavelengths. You can think of your eyes like a spectrometer in the way that it can measure wavelengths. It interprets colors as different wavelength ranges. And our spectral range is just the visible light, which is what we've defined it as. So like I said, our spectrometers are typically targeting 350 nanometers to 2500 nanometers. And our spectroradiometers are given a radiometric calibration that allows it to give energy units to these measurements. So it'll measure energy. Reflectance spectroscopy focuses on the radiation reflected by a material. So every material will have different absorption features, which will give you a reflectance curve, which is usually indicative of what the material is, what molecular bonds are going on in it, and its chemical compositions. So I'm going to go over a generalized view of what goes on inside of a spectrometer. So how the spectrometer decomposes the reflected light and how it reads from 350 to 2500 nanometers, and how our detectors convert these wavelengths intensities into electrical pulse, and our software combines all three detectors to provide the reflectance percent at each wavelength. So here at the entrance slit, the light will come into the spectrometer. It will hit the first mirror and send it down the series of dichroic beam splitters. So these essentially allow certain wavelengths to pass through them and reflects the targeted wavelengths to another mirror that then sends it to a photodiode array that is targeting those wavelengths. So the first dichroic beam splitter sends 350 nanometers to 1000 nanometers to that mirror, and then to the detector that will then measure how much the magnitude of energy on that if it has a radiometric calibration. If not, it will do it by detector counts, which then you can operate by ratios of detector counts, which is what a spectrometer does. So then the rest of the light will pass, or 1000, yeah, the rest of the light will pass through the dichroic beam splitter and hit the second one, which will target 1000 nanometers to 1900, which will send that to a mirror and then to the second detector. And then the rest of the signal will hit the last beam splitter that will send the last of the signal to that detector from 1900 to 2500. So this is important because it optimizes how much signal we're using. So we're actually using the entire signal that enters the spectrometer instead of using any moving parts, moving gratings, or bifurcated cables to bring to each detector. We're actually just decomposing that spectral signal. So we're able to get great signal to noise and using this methodology. So our instruments are designed for field use. They have no moving parts. They're thermoelectrically cooled detectors, dust and humidity proof, powder coated aluminum chassis, high impact handle and bumpers, as well as high capacity lithium ion batteries and Bluetooth connection. So this is designed for ruggedness, reliability, portability, and stability. So these instruments are designed to be used in the field, but they can be used in a lab as well. But all of these metrics were designed to make it easy to use in the field and to be reliable. So we talked a little bit about what a radiometric calibration is, but to talk more specifically, a spectroradiometer is a spectrometer with a radiometric calibration, which means it's given energy units. This allows the instrument to collect radiance and irradiance measurements that are crucial for atmospheric research, environmental science, albedo studies, as well as ground validation for satellite validation with satellites or airborne detectors. So here are a few factors that should be considered when you're collecting outdoor measurements for remote sensing. So you have your beam radiation, cloud reflection, scattered light due to the atmosphere, as well as absorbed light due to the atmosphere. And so you have diffuse radiation, which is all the light that is scattered. You can think of that as like if you're in a shadow, you can still see in the shadow. That's diffuse radiation that you're seeing. And so the light that you're getting, even if it's a clear sunny day, is still going to be affected by, if you're using the sunlight, by the atmosphere and the constituents in the air that will change. And so that's why we've measured the solar radiation. So this is a good representation, or this is a representation of solar radiation. This is what you're going to be referencing in the field. So when you take a white reference measurement with a white plate, you're trying to get the idea of a white plate is to get a representation of the total energy or light that you're measuring so that you can get a good reflectance measurement. So you take a measurement of the total light, then you take a measurement of the target, which is the light that's reflected off of an object, and that should be characteristic of the material that you're targeting. So here on the right, or on the left, is the solar radiation curve, and on the right is different materials that will absorb differently, which should all have different reflectance curves. And something else that should be noted is the two water absorption features in that left curve at 1400 and 1800. Those are water bands. You can see that there's no signal there. That's why when you're taking outdoor measurements, you'd see noise in those areas because the atmosphere is absorbing most of the radiation there. So something else that should be considered when doing reflectance measurements in the field or in the lab is specular reflection. So specular reflection is when light is nearly perfectly reflected off of a surface or a mirror-like surface. These are typically off of shiny surfaces or water. So when taking these measurements, you want to target the diffuse reflection in order to avoid specular reflection components. So if you're in a lab setting, you're going to want to be taking measurements at nadir or at a different angle at your target, and you want to avoid those specular reflected components. So those will have the same angle as the light coming down at the material reflected off the second one. So it's also important to understand the field of view that you're measuring with. So all of our fibers have a 25-degree field of view, and it will have a cone that emits out of the end of the fiber. So the distance to target is very important. These instruments are also point spectrometers, so the entire field of view is going to be averaged together into one spectra. So when you're taking measurements in the field, defining what your field of view is imperative to understanding what you're characterizing with your spectra. So this figure will be great if you want to come back to this video and use it if you're trying to calculate your field of view later. But this is something that's very important to understand when conducting field measurements. You can also change the degree of your field of view. So the bare fiber is 25 degrees. We also have 1 through 10 degree lenses that you can change what your field of view is. We also have diffusers that have 180 degree field of view. So field of view is critical, and satellite and airborne detectors are typically limited in spatially or spectrally or both. And that's why it's important to use a field-based spectroradiometer or spectrometer alongside these coarser methods to calibrate them and conduct ground truth validation. This is essentially using our high resolution to say with great certainty what is on the ground compared to other methods that may be dealing with atmospheric corrections or coarser spatial or spectral resolutions. So it's critical to use field-based spectrometers alongside these other methods to prove however you're processing your data. So our instruments are very commonly used for soil analysis. Typically, they're used to identify things like total carbon, total organic carbon, and other soil nutrients. They can also be used to identify sediment or clay mineralogies. Reflectance measurements of soils are typically conducted with either a pistol grip lens with a lens for bare soil patches or with a contact probe to provide a more targeted approach. So the contact probe has an integrated light source that will bump up your signal for darker samples. So the darker the sample is, the less that will be reflected off of it. So if it's a really dark soil, we recommend using the contact probe. Not only is it a 10 millimeter spot size that you're measuring, which is very targeted, but it also bumps up your signal to get better reflectance curves off of these materials. So you can also measure radiance and irradiance. So typically this is done with a inline diffuser. So this is a 180 degree field of view lens. So typically you would want to make sure that the inline diffuser, if you're characterizing irradiance, that you have it nice and flat and above all of the materials that you're using in the field, so that you're not measuring yourself inside of your reflectance curve. or your irradiance measurement. You don't want to be contributing to that or anything reflected off of you. So typically recommend holding it over your head and as flat as possible to get a good irradiance measurement. This is typically used for characterizing energy balances within a system, or canopy studies is very common. So taking an irradiance measurement and then going under canopy to see how much light is penetrating to the ground foliage is a very interesting study that's commonly done with this. So also with field work, something that should be noted is, like, what are your weather conditions? So ideally, every field study will be conducted with perfectly clear skies and the same conditions throughout the day, but we can't always have that. So people often ask, how often should I reference my instrument during field sampling? The answer is it depends on the weather. The more often you reference, the better, as you want to make sure you're sampling under the same conditions that you collected your reference scan, because that's what's going to define your ratio reflectance. So I recommend re-referencing after about 10 scans, but this is subject to change if there's intermittent clouds or inclement weather. So in order to combat this, we've developed Darwin Dual, which allows you to, if you have two instruments, you can use them in concert with each other to take a reference scan simultaneously with every target scan. So this minimizes the effect of changing conditions with clouds and other weather conditions. Another incredibly common measurement is in leaf reflectance measurements. This is typically conducted with the leaf clip or a pistol grip. Again, the bare fiber or lens will give you a greater average of the plant's reflectance, and the probe allows you to be more targeted. So the leaf clip's design takes the light away from the source or the target material in order to minimize any damage that the heat of the light source would do to that leaf. So for even more targeted approaches, you can also use the pine needle adapter, which lets you get down to the millimeter scale or the pine needle scale reflectance measurements. But the leaf clip emphasizes the non-destructive nature of this sampling and makes sure that you can take continuous measurements on the same leaves without drying them out. So this is important for a lot of different plant nutrient metrics or plant health metrics or biodiversity as well. So you can take these spectra and bring them into EasyID and actually conduct speciation based on the reflectance, because most plants will have either different chlorophylls or different cell structures that you can see within the reflected spectra. So speciation and biodiversity, as well as invasive species studies, are very common with this kind of measurement. So you can do that with plants. You can also do that with algae and different ocean biomes. So water quality measurements are very common. You can conduct algae bloom monitoring, speciation of micro and macro algae. You can define different suspended sediments as well as CDOM and many other metrics. So with water reflectance measurements, it's very important to avoid those specular components, because the ocean surface is very shiny or mirror-like, especially if it's a very calm day. So it's important to define your angle with the sun and make sure that you're not collecting data directly towards the sun. There's many different procedures for this. For a good procedure to collect water reflectance measurements, NASA's Delta X program used our PSR 1100F, and they collected scans that were used to calculate remote sensing reflectance, water leaving radiance, relative to downwelling irradiance. And these will often include a correction for the influence of reflected light. So this is an example of some spectra taken off of a lake. And so you can see something that should be understood when taking water reflectance measurements is the magnitude of energy that's leaving the water is significantly lower than any other surface. The water absorption is very strong. And so you might be measuring on like under 5% reflectance on some of these, or even under 1% reflectance. But as you can see in this curve, we were under 1% reflectance, and we're still getting clear data here. So there's a lot of different indices that you can use. Darwin will automatically calculate. So we have 19 integrated vegetation indices like NDVI. It will collect all of your metadata when sampling. And then you can send all these metrics into EasyID, where you can do spectral matching. Darwin also has the ability to conduct a lot of different spectral math and in order to develop procedures with data processing with your data and look at them in derivative format as well. So, like I said, reflectance measurements are commonly used to do identification of materials. So this could be mineral identification for vectoring resources, as well as different soil mineralogies or nutrient content, as well as vegetation, speciation, and nutrients and health. So it's very common to tease out the spectra and figure out what material is being scanned based on the reflectance. So remote sensing is a diverse field. It requires an instrument that can be used in many different research areas and can be adapted to nearly any remote sensing applications. Fundamentally, Spectral Evolution's emphasis on spectral resolution and signal noise allows our instruments to be used alongside nearly all modern remote sensing satellites and cameras. And our instruments are ideal for nearly all of your field reflectance spectroscopy needs. So I'm happy to answer any questions, and thank you very much for your time.